Autophagy, the cell's ancient recycling system, is both guardian and accomplice in the story of cancer. At its core, autophagy protects normal cells from stress, clearing away damaged proteins, defective mitochondria, and toxic debris that could otherwise spark genomic instability. In this way, it prevents the earliest seeds of malignancy from taking root. Yet, as cancer develops, this same mechanism often turns traitor. Once a tumour is established, autophagy no longer acts as a defender, but becomes a supplier, feeding the growing mass with recycled nutrients. In nutrient-starved tumour environments, autophagy helps cancer cells survive low oxygen, scarce glucose, and the stress of therapy. The very system that once maintained balance now fuels persistence. Researchers have discovered that many cancer cells manipulate the epigenetic landscape to control autophagy. Histone methylation and acetylation, chemical marks that turn genes on or off, can silence or awaken autophagic genes depending on the cell's needs. When the EZH2 enzyme represses autophagy, tumours gain an advantage, but drugs that block this enzyme can reactivate the cell's cleaning machinery, forcing cancer cells into self-destruction. Evidence from genetic studies strengthens this paradox. In several human cancers, breast, ovarian, prostate and liver, key autophagy genes like BECN1, ATG5 and UVRAG are often deleted or silenced. In animal models, restoring these genes reduces tumour growth, showing that functional autophagy can act as a brake on malignancy. At the same time, tumours exploit it to withstand chemotherapy, radiotherapy and metabolic stress. Within the tumour microenvironment, autophagy influences more than cancer cells themselves. It reshapes the behaviour of endothelial cells, fibroblasts and immune cells, each adapting to the nutrient-depleted landscape. In endothelial cells, autophagy allows survival under hypoxia, but also drives the abnormal vessel growth that nourishes tumours. This creates a vicious cycle. As cancer consumes, autophagy ensures it never starves. Modern research has revealed that autophagy interacts intricately with other death pathways such as apoptosis and ferroptosis. It can delay apoptosis to protect tumours, or in other settings sensitize them to ferroptotic death by degrading ferritin and freeing iron. This flexibility gives autophagy its dangerous versatility, both a survival shield and a potential therapeutic target. Ultimately, autophagy embodies the duality of life itself, a process meant to preserve order that, when hijacked, sustains chaos. To treat cancer, scientists must decide when to awaken this system and when to silence it, a delicate balance between letting the cell recycle or forcing it to decay.